morning, everyone. Welcome to breakfast at UMHealth. Today we have two very young and prominent speakers. I will introduce the first one, Dr. Arutchelvan Rajamanikam. He is a researcher and senior lecturer from the Department of Parasitology, Faculty of Medicine, UM. Dr. Arun has been actively involved in the research on understanding the interactions of protozoan parasite, mainly Pathocystis species, with its environment and health and disease individuals. Arun completed his undergrad studies in the field of resource biotechnology in 2010 and ventured into parasitology during his master studies in UM. His interest in understanding the interactions and synergies of intestinal parasite and gut microbiota led him to complete his PhD. He currently aims to decipher the gut microbiome diversity patterns, metabolic profile, and function in understanding health and explore the predictive ability of the gut microbiota for the benefit of preventive medicines. He also worked actively to understand the intricate bacterial-parasite interactions in various protozoa parasite infections. Today, Dr. Arun holds funds close to 1 million from international and local funders such as UKRI, FIGS, PKPUM, SPI, and CORE-PI. He has contributed immensely as reviewer of various ISI index journals, and recently he has appointed as a member of European Cooperation in Science and Technology Actions for Pathocystis species in UMHealth. Apart from academia, he also contributes in various national policies, making efforts as an affiliate to the Young Scientist Network ASN. Let us welcome Dr. Arun to begin his talk.
Thank you, Prof. Lau. So I believe you can see my screen, right?
Yes.
Okay, it's the full screen, huh? It's not the...
Yes, it is.
Okay. Yeah. Okay, I figured that out. Okay. All right, very good morning. It's really good to see all of you in the breakfast at UMHealth session. I believe all of you all have had your breakfast, or at least having your breakfast while you're listening to me. You see, most of the time when there is talk on bacteria, you will see that researchers who are giving the talk, they will come on from a couple of maybe different departments. And the same goes for virus. You know, people will be researching on virus from various departments. But when it comes to parasites, you will only see people coming from only parasitology. And worse so is research on intestinal parasites. There is just a couple of labs that's working, and mine is one of it. So today, we are rare PISA. So we have another unique and rare study here that we would like to share with you. So the question, can a harmless organism be turned harmful? Role of gut microbiota. So that's what we are going to explore today. So before I can start, let me bring you to the terminologies that is often interchangeably used, the microbiome and microbiota. So many people use it interchangeably, but without knowing the actual meaning. So microbiome refers to the genes of cells and viruses. They don't refer to the cells by itself. And microbiota is the reference to the cells and viruses. So with that clear, let's dive deeper into what is gut microbiota is all about. So gut microbiota is a consortium of bacteria, virus, fungi, and protozoa that live in peace, in harmony between each other. And they are mainly shaped by some environmental signals and diet, most primarily, but they are also shaped by various other factors. And this microbiota contributes to various metabolites. It can be LPS, bile acids, and so on and so forth, which then affect or influences the system, various different systems in human body, and that leads to health or disease. About one thing that is often underlooked in the arena of gut microbiota is the single-cell eukaryotes, primarily protozoa. And when we say protozoa, they are primarily consist of parasitic protozoa. And 90% of them are often asymptomatic infection. They are there, they colonize happily, but they are asymptomatic infection. The question is, why is that? Why do we see asymptomatic infection when these are parasitic protozoans? So most study at the moment, although it's inconclusive, finds that there is a balance that the parasites strike with the microbiota so as to not cause any severe damage to the host. So the current finding, there are a lot of contradicting outcomes there. For example, some study says the gut microbiota showed no changes despite there is parasitic infection, and some says there's increased diversity due to parasitic infection. There's quite a bit of research that has said this, and quite a number of studies have said that reduced diversity, but exactly we don't know what's happening. And the influence of parasite genotype is unclear because parasites, the protozoan parasites specifically, have got a wide genetic diversity, and they may contribute to various genotypic outcomes. And the impact on gut parasite environment on parasite is also not clear. So that's what we are focusing here. So we need a model organism to actually study this further. So that's why we picked blastocystis. And blastocystis is a protozoan parasite that is associated with non-specific gastrointestinal symptoms. And more than 1 billion individuals were estimated to be infected in 2013. And now we are 12 years ahead. I believe the number would have definitely increased. And the prevalence is definitely higher in developing nations. And some countries have reported up to 100% prevalence in Senegal, for example. And the interesting thing is it colonized both symptomatic and asymptomatic individuals, making them commonly associated with commensalism as well. And they are often associated with non-specific gastrointestinal symptoms and skin rash. Some of the studies have indicated skin implications on skin rash, IBS, as well as colorectal cancer. However, the mechanism and causality are not conclusive yet. So these are the morphological forms of blastocystis. The cyst form is the transmissible form, and then it infects the human, and there will be various different forms in the host. But the question now is, what is its interaction with the gut environment? So we started off with our natural experiment that was already happening. So we compared blastocystis isolated from orang asli population and urban population. Both of them were blastocystis subtype 3. This is mainly for genetical standardization, and we isolated from both symptomatic and asymptomatic individuals in both this population. And we found that they had some difference in their growth profile and generation time. And there was also some difference in terms of their apoptotic forms that they form. See, this one I have to stress here that apoptotic forms in blastocystis has been referred previously to contribute to increase in cell numbers in vitro. So we tested that. We found that there was difference between urban and orang asli. And the morphology in terms of transmission electron microscope was also different. We found that certain asymptomatic isolate, like urban asymptomatic isolate, has obvious fuzzy coat seen compared to orang asli symptomatic and asymptomatic isolates. And the orang asli isolates, blastocystis isolated from orang asli, were able to withstand higher drug concentration. That means they are more robust compared to the isolates from urban. And see, to make it interesting, it was not only in terms of its morphological features, even in terms of its antigenic features. So we normally, those who are working with parasites, they know that proteases are often regarded as the virulent factor in parasitic infection. And when we compared the urban and orang asli blastocystis, we found that there was completely different profile of protease seen, and these proteases were able to differently proliferate cancer cells, which is often a marker to identify the pathogenicity of parasite. This was then published some years back. So the question was, okay, what if that they were two disparate communities. I mean, urban and orang asli, we know that they are so different, their lifestyle is totally different. And therefore, it could have caused that the parasite to be, it's probably different parasites that are there. So we tested again only on urban population. We compared those individuals of a blastocystis from schizophrenic individual as well as normal. And we found that those blastocystis isolated from schizophrenic individuals had greater drug resistance, which is similar to what we saw earlier. And they were also exhibiting a different protease profile. And this was published a couple of years back. So this means that a blastocystis, when it's exposed to a different gut microbiota, let it be a non-schizophrenic or schizophrenic gut microbiota, may exhibit different different phenotypic features, and that may lead to a different clinical outcome as well. So to test that, we went on to look at the gut microbiota. So the question now that we carry is there emergence of different variants of blastocystis? Could that lead to a pathogenic and non-pathogenic variant? So we collected samples from 50 individuals who was asymptomatic and symptomatic, and within each asymptomatic and symptomatic individuals, they were blastocystis positive and negative, and we assessed the gut microbial diversity to see how diverse was the microbial composition, and whether a specific bacteria or a couple of bacteria which was differentially abundant as a result of blastocystis infection, and whether there is the functional prediction, whether the functional aspect of the microbiota has changed as a result of blastocystis infection in both symptomatic and asymptomatic. And we found that, yeah, of course, the microbiota diversity has significantly changed. If you see in asymptomatic, those with blastopositive, the microbiota was significantly reduced, but there was no much, the diversity was significantly reduced, but there was no much difference seen in symptomatic, whether it's positive or negative. And if you look at another plot, which tells us about the diversity, I want you to just take a look at the yellow eclipse here, which means this yellow eclipse is actually blastopositive symptomatic, isolated from a symptomatic individual. So this blastopositive symptomatic individual has a unique microbiota profile, which is dissimilar to other eclipse. So that tells us that when it is symptomatic and there's blastocystis there, that means the microbiota is different. So is the microbiota is causing the parasite to behave differently, or it is the parasite which changes the microbiota? It's something that we have yet to answer. And we looked at what are the bacteria that could be differentially abundant, and we found that, you see, in symptomatic and asymptomatic individual, there was totally different bacteria that was found differentially abundant in blastopositive individuals. So that again reaffirms that there is something else taking place. And we looked at the functional prediction, and we found that the top plot, which is symptomatic, and the bottom plot, which is asymptomatic, the top plot, both of that shows that in blastopositive and in symptomatic and asymptomatic, there was difference in terms of its function. Now the question is, if that's so, what will happen if I change the accompanying bacteria? So now we see that the bacteria in symptomatic and the bacteria that is accompanying blastocystis in asymptomatic is different. The evidence is there already. Now, what will happen if we change this accompanying bacteria? So what we did was we took this was done entirely in vitro. So symptomatic blastocystis were cultured, and they were then co-cultured with a bacterial suspension from asymptomatic blastocystis culture. And likewise, we took asymptomatic blastocystis isolate and co-cultured with bacterial suspension from a symptomatic blastocystis isolate. And what we saw was the blastocystis, which was initially symptomatic, behaved like asymptomatic, and likewise for asymptomatic isolates. So here are the evidence. So this is asymptomatic and symptomatic graft, and upon co-culturing with different bacterial suspension, they behaved like, say, for example, symptomatic isolate. When it's cultured with bacterial suspension from asymptomatic isolate, when it's cultured with bacterial suspension from symptomatic, they showed a growth profile which is similar to symptomatic isolates. And likewise, we saw for symptomatic isolates as well. And to our surprise, this was not only seen phenotypically, we also saw in terms of its antigenic content. Although symptomatic isolates, when it was cultured with asymptomatic bacteria, there was no much changes. But in asymptomatic parasite, when it was cultured with bacteria from symptomatic blastocystis, we saw the serine proteases has shifted to cysteine proteases. This is quite important because cysteine proteases are a type of protease that is often implicated with as a virulent factor in various protozoan parasitic infection. So here we are making an important note here that the bacterial changes may influence the parasite, and this can cause the parasite to produce a different protein. Now, coming back to our question, can a harmless organism be converted into a harmful organism? In the case of blastocystis, based on the evidence that we have seen, it is likely, but we are not sure how yet. So this is highly contributing to our understanding of so much of hypervariation in blastocystis infection, or in mostly in parasitic protozoan infection. And that is probably why 90% of the cases are asymptomatic. It's because of the gut microbiota, which is contributing to the parasite. Guess what? This is not a new science. So those two scientists, Casadeval and Pirovsky, in 2003, they proposed a framework and they called it damage response framework of microbial pathogenesis. So these two scientists are from Johns Hopkins, and the other one, I think Pirovsky, is from Albert Einstein School of Medicine. And what did this framework say? So this framework says that pathogenicity is not a microbial trait, but it's an outcome of interaction with its host environment. And that's exactly what we have seen here. But we have got some evidence, but we need a lot more to be done before we can say this conclusively. So our future studies have now shifted from a reductionist approach to a more system thinking, looking at the interaction. And we have now some interesting study which was currently being funded internationally as well to develop axenic model of blastocystis, which is the model organism that we have used to integrate the role of bacteria on protozoan parasite. And we are looking at parasite bacteria interaction and its implication in colorectal cancer aggravation. There's a student working on this. And parasite bacteria interaction on drug response in intestinal parasites. There's another research that's ongoing on this aspect. So having said all that, understanding the fact that gut microbiota is playing a crucial role apart from all the other roles that we know medically. Now, this is another role that has been added under the belt of gut microbiota, which is its influence on other protozoan parasitic infection that can sometimes be asymptomatic or referred as commensal. So the challenge now, the most difficult challenge now is guarding the gut, which is maintaining a healthy gut. So at this point of time, of course, there are also other ways that you can maintain a healthy gut. One of the biggest challenges that we are facing currently is getting a healthy gut with a good diet. So with that, if you have a healthy gut, then potentially we are not going to expect much changes because healthy gut is often referred to as a stable gut. A stable gut will not undergo much changes. Therefore, there will not be such instances that we see as I said earlier. So I would like to acknowledge my research team, which is led by Prof. Suresh and Dr. Chandra, and also all the postgraduate students. And with that, I think I've made my point. Thank you.
Thank you. very much for a very interesting talk, Arun. Let me see the chat box so far. There's no question so far. So, Arun, what do you think about the future of your research? So, what else you can do more with this?
Yeah, there's a lot more actually that can be done at this moment because we have whatever we have did is without the inclusion of an exenic model. That means we don't have blasto only culture. So all this while all our cultures is with the parasite growing in bacteria in which we purify them and then introduce change the bacteria and all that. So that is one thing, and we do not know whether they are responding to any specific bacteria. Let's say, for example, if there is any particular pathogenic bacteria like Klebsiella or Acinomycter baumannii or maybe Pseudomonas aeruginosa, how are they responding to this? That
Mm,
we have.
interesting. So, there are actually three questions in the chat box that I missed just now. The first one is by Dr. Chan. The presentation was most interesting. Is the profile different between vegetarian and what non-vegetarian?
Yeah. Definitely the profile will be different. It has been established in various other studies, but not in my study. We did not look at the dietary profile of those individuals. But I have seen quite a number of studies who are vegetarians. They have a more diverse microbiota compared to non-vegetarian. So it's always encouraged to have a lot more vegetarian diet, which is difficult to get in our faculty. Definitely.
Interesting. So another question by Dr. Chan as well. Is the profile different for those with large volume stool compared with those with low volume? The stool
Uh,
volume.
is the profile different for those with large volumes too compared to
Mm,
those with low volumes too?
mm,
Okay, I'm so sorry. We have not actually tested this, but this is something very interesting to see actually.
I see.
Yeah.
Okay. And then one more from Prof. Musara. After taking antibiotics, do you encourage probiotics?
Oh, yes. Yeah. So
Mm,
because antibiotics is something that is non-targeted, it also affects the non-targeted organism, which is primarily in the gut. So which also can contribute to significant changes. And comparing to our data in the schizophrenic individuals, most of them they have altered gut microbiota. I did not include those data here. They have altered microbiota because of their
mm,
frequent usage of psychobiotics and psychotic medicine and also antibiotics. And that is contributing to resistance in parasite in against drug or robustness against drug treatment in this kind of parasitic organism.
mm. I see. How about those having probiotics frequently? I'm sure that affect the microbiota as
Yeah, yeah. So probiotic have been seen because we
well.
saw that in our survey that those who predominantly take probiotics, their likelihood of developing schizophrenia was much lower. So probiotics that they consume and was mainly on yogurt and this kind of probiotics.
Mm.
So I believe that these probiotics do play a role here.
So there are two questions from Prof. Suresh, who is the expert. So how to establish a healthy gut? I think you need another lecture on that.
So yeah, establishing a healthy gut is something Prof. Suresh has spoken before, which is ease, exercises, eating healthy, maintaining a positive attitude, and I don't know what is S at this point of time. So there's and also there's mainly it's the diet. Yeah, mainly it's the diet, and also it's when do we eat, and also how do we eat. Those are the two important aspects. And yeah, I think mainly that.
Okay, good. He's also asking what evidences in the future will you look for to establish the questions that organism can interchange their pathogenic abilities.
What evidences in the future? So
Hmm.
the future evidences that we are keen to look at is mainly in terms of their phenotype, whether they are exhibiting certain phenotype that can that has been associated to pathogenic ability in the past, like the surface can change, whether the surface changes is associated to pathogenicity, or certain proteins or certain enzymes that they're secreting, which can actually contribute to, because protease is a very general protein. It's very difficult to actually compare them, but specifically for that organism, is there any pathogenic molecule that has been linked with the environment the environment influencing that?
That maybe you need metabolomic studies.
Yeah, exactly. Yeah. So we need the exenic model, which we have successfully developed at this point of time, but we need to validate and so on and so forth.
That's good. Prof. Tunggu Sarah would also like to follow up with the probiotics. What kind, what kind of probiotics do you encourage? Any fermented food?
I'm not a probiotic expert here to actually prescribe, but most encouraged is yogurt. But there are also some people who are sensitive to yogurt, and also the best kind of food to include to improve the gut health is prebiotics. Prebiotics are the food that we take in, which is not digested by our human body, but it is a source of food for the bacteria in the gut. And prebiotics is everywhere in the vegetables, in the lentils, and all the kind of food that we eat.
Right.
For processed food.
Okay. Last questions from Dr. Chan. It's not a question actually. He's saying that it would be nice if you can look into the sequence of time instead of one time point.
Yeah, that's right. That is one of the confounders that we have right now, because we looked at point in time. But what we are now working now, is we are going deeper into whole genome sequencing for a longitudinal aspect of gut microbiota with blastocystis infection. Yeah.
Right. Okay.
Dr. Quentin.
All right. I think we covered all. Very well done, Arun, and we are on time. Since we are rushing, I think we need to wrap up this session today. Thank you everyone again for joining us. I will pass the floor to the next speaker.
Thank you.
Thank
Prof.
you.
Thank you.
Thank you.